Hi CSBL members. The title of journal I'm going to introduce is Engineering High Efficiency Matriptase Substrates Using E. coli Display for Applications in Pro-Drug Activation, which published on Cell Reports Methods in June this year. This article introduced to develop an E. coli display platform for high-throughput screening of protease substrates. Using this method, they identify matriptase cleavable sequences that outperform existing substrates and enable efficient prodrug activation. Here are the highlights of this article. In this study, high-throughput E. coli display enables the screening of millions of protease substrates. Among them, top hits show more than 40-fold improved specificity constant over previously reported matriptase substrates, and they also optimized the substrates enable efficient antibody prodrug activation in vitro. So what is prodrug? A prodrug is a compound with little or no pharmacological activity that metabolizes inside the body and converts into a pharmacologically active drug compound. Prodrugs are designed to overcome pharmaceutical, pharmacokinetic, and/or pharmacodynamic challenges. Here are the core components. First, payload, the active drug or bioactive binder. Second, masking or promoting, blocks the drug's activity or binding until removed. Third, trigger or linker, a cleavable motif that responds to a specific cue. Fourth, targeting moiety, directs the prodrug to diseased tissue. And what is matriptase? Matriptase is a type 2 transmembrane serine protease encoded by ST14 gene, essentially tiny molecular scissors embedded in the membranes of epithelial cells in tissues like skin and gut. It is made as an inactive zymogen and tightly controlled by natural inhibitors such as HAI1, HAI2 to maintain normal barrier function and tissue remodeling. In many epithelial cancers, however, matriptase becomes dysregulated. Its expression and activity rises near tumor cells, where it can trim extracellular matrix proteins and activate signaling molecules that aid invasion and spread. Because its catalytic site faces outward and shows a clear sequence preference, researchers can design peptide linkers that matriptase cuts efficiently. This makes matriptase a useful switch for prodrugs. Therapies kept inactive in blood that are amassed only after encountering active enzyme in the tumor microenvironment, improving precision and potentially reducing side effects. The aim of this study was to identify new matriptase substrate sequences that could be cleaved more efficiently compared to previously reported substrates. The display method is based on the expression of recombinant proteins on the outer membrane of E. coli using a genetic fusion to the outer transporter IDA as shown in Figure A. A plasmid called p-PALU-CRS derives an IDA outer transporter fusion that places 3 parts in the series on the E. coli surface. An affibody as a cleavage reporter, a variable protease substrate in the linker, and albumin binding domain ABD as an expression reporter. It's arabinose inducible and includes the signal peptide. The fusion is multivalently displayed on the outer membrane by IDA outer transporter, exposing the substrate between affibody and ABD for on-cell protease access. After protease treatment, cells are stained with HER2 biotin SAPE to read the cleavage reporter and SHA Alexa 647 to read the expression reporter. Non-cleavable substrates keep both signals, like gray line shown in Figure D, and cleavable one both the HER2 signal, the red line. The study validated an E. coli surface display vector using substrates for matriptase as well TEV protease as control. Upon treatment with the matching protease, cells showed reduced fluorescence from the cleavage reporter, confirming specific on-target cleavage. No cross-cleavage was observed between the two proteases. Dual labeling of expression and the cleavage reporters indicated minimal static hindrance on the cell surface. Mixing the two display populations one-to-one and treating with a specific protease demonstrated that VAX can be effectively used to enrich substrate populations with diverse cleavage properties from combinatorial peptide libraries displayed on E. coli. The E. coli displayed substrate library was split and exposed to matriptase or PBS control, then labeled and analyzed by flow cytometry. Initially, most variants were not cleaved, but after 4 rounds of VAX, progressively enriched for cleavable linkers. Even the concentration of matriptase reduced from 50 to 10 nanomole to raise stringency, the result evidenced by increasing loss of the cleavage reporter. By the final sort, the library was dominated by matriptase cleavable substrates, whereas PBS controls showed no detectable cleavage. The libraries were compared to evaluate the most enriched clusters, the amino acid distribution at each position of the substrate is shown in Figure B, with percentage indicating the frequency of each amino acid in these positions. In the original library, positions P3 and P2 were fully randomized. After sorting, trend showing enrichment for amino acids with the longer side chains at P3, like methionine, lysine, and arginine, and the small side chains at P2, alanine and glycine. Negatively charged amino acids were largely disfavored across these positions. On position P1, both arginine and lysine were retained, but there was a preference for arginine over lysine. Additionally, positively charged amino acids, especially arginine, were enriched in other positions, suggesting that the cleavage site may have shifted in some substrates. In position P1', a similar preference for arginine over lysine was observed. Although P2' was designed with a preference for small amino acids, this tendency was less obvious in the enriched clusters. The data indicated a general preference for hydrophobic P3' amino acids, in line with the design of the original substrate library. Researchers screened 192 post-salt E. coli clones by flow cytometry and chose 7 substrates for detailed testing, and their sequences are listed in table. Across 1 to 50 nanomole matriptase, 3 candidates, B9, C5, and C9, were top performers. Even at 1 nanomole, over than 50% cleavage by 90 minutes, and completely cleaved at 10 nanomole or 25 nanomole. The top substrates B9, C5, C9, and the controls were synthesized with ABZ-DMP FRET labels and assayed with serial dilutions of substrate for 90 minutes. The FRET kinetics agreed with the flow data, identifying C5 as the fastest substrate. The specific constant value for C5 was 40-fold higher than that of the previously reported matriptase control substrate. And these substrates were also used to assess stability in the presence of various proteases. The substrates were incubated for 100 minutes with cysteine proteases, serine proteases, and matrix metalloproteases, and no detectable substrate cleavage was observed with any of these proteases, shown in Figure A to C. In contrast, cleavage was observed when the substrates were incubated with matriptase under identical conditions, shown in Figure D. The authors inserted the 3 best substrates into antibody products and compared them to an matriptase control and a non-cleavable TV control. Products were incubated with matriptase for 10 to 60 minutes and analyzed by SDS page. Distinct bands corresponding to intact prodrug, heavy chain plus-minus mask, light chain, and released about 7.5 kilodalton mask confirmed cleavage products. And the band intensities from previous SDS page were quantified in ImageJ to calculate percent cleavage. At 1 nanomole matriptase for 30 minutes, the new substrates achieved 24 to 35% cleavage, while the control showed none. At 10 nanomole for 30 minutes, all 3 new substrates were fully cleaved. The control required 50 nanomole for 60 minutes to reach only about 20% cleavage. Across conditions, the optimized linkers enabled faster, more efficient activation than the legacy control. This data validate that short treatment times and low matriptase levels can activate the new prodrugs. In this study, authors built a quantitative equalized surface display and effect system to engineer matriptase cleavable peptide linkers for tumor-selective prodrug activation. They screened for more than 4 million variant libraries through 4 increasingly stringent FACS runs, enriching true cleavable and mapping position preferences by deep sequencing. Notably, the most efficient substrate, C5, showed 40 times higher than the reference substrate, indicating significantly enhanced cleavage efficiency. Inserted into antibody prodrugs, top linkers achieved 24 to 35% cleavage at 1 nanomole and completely cleavage at 10 nanomole, far outperforming the legacy control. The workflow rapidly ranks substrate and provides quantitative validation, yielding a tunable toolkit for protease-activated prodrugs and diagnostic pending application-specific stability and selectivity testing. There are also limitations exist in this study, such as display bias. In vitro screen may not fully recapitulate to the tumor microenvironment. Detailed kinetics still require follow-up and biochemical assays, and the library was placed to non-matriptase preferences, potentially limiting discovery of unconventional motifs. This is the end of my presentation. Thank you for your listening.